Thank you so much for having me. I love this conference. Love being able to see all my families here as well. What a special experience. I think one of the most amazing things about being part of this community is the rich depth and breadth of where we are in our journeys. I think it's really important to learn from each other. We do not have to reinvent the wheel. And so today I'm going to do an overview, really what's out there and what I think is on the horizon, trying to keep it as broad as possible because I know each one of us is passionate about a different leukodystrophy or leukodystrophies. I do have a couple disclosures. I work at the Children's Hospital of Philadelphia, where I'm a physician scientist. I'm a child neurologist. I am a research consultant for Orchard, Takeda, Biogen. I have several forms of NIH funding, and I do have funding from several family foundations as well. So we're all here because we're passionate about rare diseases. And this is kind of to my point. We need to unite together because while each one of our disorders are individually rare, collectively, we have huge voice. We think that there are 7,000 rare diseases, and up to 25 million Americans are affected by them. But very, very few, less than 10%, have any treatment options at all that are targeted. So we have a lot of work as a community, but again, we don't have to reinvent the wheel. We can see the paths that our friends and colleagues have traveled and be able to learn from their experiences. Most genetic disorders that are rare are single gene diseases, which is the category that almost all of the leukodystrophies fall into. I wanted to really briefly, for the families in the room, give an overview. Normally when we talk about moving to clinical trial, we just talk about the clinical trial parts, which is the discovery and research. So in the bench, we screen compounds, we develop new gene therapies, so we try and see what works. We then move into preclinical research where we try these potential treatments, oftentimes in animals. We then have a phase 1, which is a safety trial. That has usually very small numbers. A phase 2 trial looks at both efficacy and dosing. What's the right amount to help families? And in phase 3, we look at efficacy, and we always continue looking at safety. At this point, in the traditional schematic, drugs go for regulatory review, where the FDA or whatever your local regulatory body reviews this, and then you go into a phase 4 trial, which we don't think about a lot, but what that is it's post-marketing. Once something is approved, you still need to make sure and follow it's safe. But actually, if you look on the left side, we do a lot of stuff before we get to this clinical trial. It's not just what happens in the bench, it's the natural history studies. It's working with families and registries to identify potential patients, understand how the disease affects them, identify what's important to families and how we can best measure it, and determine biomarkers or things that we measure that can help us see, either predict the future or see if a treatment is getting to where we want it to. I think we're blowing up the system because now we're starting to develop treatments for rare diseases. And this traditional kind of linear flow where you're hoping to get hundreds to thousands of people in your clinical trial just isn't possible. And so one of the things I want to emphasize is the importance of us being really certain of what the natural history is of our rare diseases before we go into a clinical trial. So I put up a fake study here. So let's say we have a hypothesis, and you always in a clinical study have to state your hypothesis first. You want to say, I think I'll have a 50% improvement, which means turning purple. And you have a baseline enrollment. Here, you enroll 3 people, and at year 1, only 1 turns purple. Okay, your trial failed. You might have enough suggestion that you can do the trial over again, but that 33% improvement doesn't count. So let's say you can extend it longer, and 1 person turns purple at 1 year, and everyone else turns purple at 2 years. This means the study had a lot of flaws. You have multiple populations that have different journeys or trajectories in your initial enrollment cohort, and maybe you didn't study long enough. Maybe I should have asked that same question, but given a little bit more time to follow my population. So imagine again, Baseline enrollment, only 33%. But what if I develop a biomarker, either imaging or something I can measure, that helps me sort into 2 different groups? And now I can have one group I'm seeing if it gets a change at a year, and another one at 2 years. And so what we're looking to do is develop clinical trials that both are as narrow and defined as we can, but have something we call generalizability, which means it can help the population outside of just the children that were studied. And that's the tension we have when we're trying to develop a clinical trial. There are lots of missteps. And again, we're going to learn from our friends that have gone down this journey already. One problem is sometimes a drug works really well in a petri dish, but when we get it into the patient, it's not getting to the cells that we needed to get to. Maybe we didn't understand the science behind the disease well enough. Or maybe we gave it as an oral medication, and that just didn't get through the blood-brain barrier and wasn't able to reach the brain. As we start doing clinical trial development, sometimes, as I illustrated, we choose too broad of a group, and we asked a question that was a great question, but we had the wrong timing, or we weren't able to predict well enough. And sometimes we choose a goal that's too hard to measure. Or maybe we thought one thing would change, but it didn't matter to the patient community, and we didn't ask them ahead of time. And so it's getting the patients involved very early to help us figure out what's important to them, and then we design the trial around that, not vice versa. Because we have a mixed audience here, I'm going to briefly go through kind of the basic science of genetics so that when we get into the therapy development, we're all on the same page. So bear with me if you're an expert in genetics. So back in 1865, Gregor Mendel first started talking about heredity. The concept that we could pass down changes from one person to another arose. And, 20 years later, we figured out cell division and how that information was changing hands. And now we have zipped forward in the intervening time to really understanding on a very detailed way where DNA, which is kind of that instruction manual in our cells that tells our cells how to make every single essential piece, gets turned into mRNA, and we now have mRNA targeting therapies that then gets converted into a polypeptide through translation and turned into a protein, and we have protein replacement therapies as well. When we're talking about genetic disorders, we're talking about variants. What these are is they're a spelling change that changes that instruction manual. And sometimes that means the protein is made in a different way, and sometimes it means the protein isn't made at all. And so we can intervene at any one of these steps. We figured out how to change the DNA instructions, how to change how that mRNA is processed so we can target the RNA, and we have figured out for some disorders how to target the proteins. And so if you can remember those 3 basic buckets, we know how in some diseases, and we're working out the kinks on how to target each one of these levels. So we're going to get into the interventions. We're going to make this as interactive as we can, so if people have questions, you can ask me. This is the schematic of what we're going to talk about. So if someone is missing an enzyme because their instruction manual didn't give them the instructions on how to make the protein, and usually this is an enzyme, we can just give it. I'm not giving you the instruction manual, I'm just giving you the missing piece. Enzyme replacement therapy has been used for a long time, especially in lysosomal enzyme disorders. Gaucher, Fabry, MPS 1, 2, 6, and Pompe disease all have enzyme replacement therapy. Okay, what if we want to step it back and we want to give something, maybe we'll have a longer lasting information, not just giving the piece that's missing, but we're going to give the instruction manual. That's what gene therapy does. And honestly, that's what bone marrow transplant does as well. And there are several gene therapies targeting the leukodystrophies and neurogenetic disorders that have been approved or are in various stages of almost approval. So metachromatic leukodystrophy has approved gene therapy in Europe. Adrenal leukodystrophy is clinically available in the United States now. SMA has gene therapy as well. So again, this isn't giving just the piece you're missing, but giving that important instruction manual. We also, through changing the RNA, that's that middle step, we can change how the instructions are carried out. Right? So we can modify that through things like antisense oligonucleotide therapy that targets the RNA level. Drugs like Spinraza, which is for spinal muscular atrophy, change that instruction manual because it's just a different way you're going to make your protein. We can also kind of ignore that entire pathway and help way downstream. So things like the recently improved Friedreich's ataxia drug work downstream. That affects reactive oxygen species and inflammation. It didn't change that instruction manual, it changed 20 steps down the road. We also can kind of change that circuitry, and we're going to talk a little bit more in depth about the supplementation we can use in CTX, which is the leukodystrophy. It isn't giving new instructions, it's just giving the piece that's missing in a different way. That one is not FDA approved, though. And then there's something called gene editing. So unlike gene therapy, where you're giving a second instruction manual, the second instruction manual helps you make the missing protein, you can actually go in and fix the original spelling error, and that's called gene editing. That's used in sickle cell disease and is really a hot new area of investigation for rare disease research. So in brief, supplementation therapy is when you're missing a piece, and we can give it. So in the case of CTX, and we heard about this on the scientific day, as well as I believe they're speaking today, in CTX, we're missing the ability to make something called CDCA. And when your CDCA is missing, it backs up the system. And it's actually the toxic backup and that feedback loop that tells the body you don't have it that is causing so much trouble. And so those toxic metabolites start building up bigger and bigger. So what happens if you just give that missing piece? Then the system realizes it's there. And it doesn't matter if it's not processing it or making it in a normal way. It doesn't cause that backup to happen in the system, and it works really well. As I mentioned, you can also sometimes give missing enzymes especially. We're quite good in general at making enzymes and doing infusions. Again, many of the lysosomal storage disorders have the ability to give a missing enzyme. And it can supplement expression, meaning say your enzyme is here, you need it up here. And so you can get either weekly or monthly infusions of the missing enzyme. This has been explored in the leukodystrophy field. We don't have any approved therapies yet, but the Shire trial looked at infusing by IV the missing enzyme for metachromatic leukodystrophy, and they have an ongoing trial actually infusing it intrathecal, which means around the spinal cord area, to see if we can give the missing enzyme for MLD as well. We can also do supplement-based innovations. And so this is where we're going to talk a little bit about gene therapy, and I know there's another meeting about gene therapy. The general premise is with bone marrow transplant, you can either harvest hematopoietic stem cells or mesenchymal stem cells, and you can give them to someone else that's missing that important piece of information. Those two fuse together and that forms a chimera, which means you have your original cells and you have a little bit of someone else's cells, either the stem cells or the mesenchymal stem cells. This has been used quite effectively in adrenal leukodystrophy, although there's much more mixed results for Krabbe and MLD, which is why we're working so hard on additional gene therapy approaches. The problem also with stem cell transplantation is you have to have a match. Not everyone actually has a match, and so there is inequities in access to care. That within the Caucasian population, your probability of having a match is actually quite good. It's around 80%. But if you're a minority, your chances of having a match could be as low as 20%, which is why even if transplant works, we still need things like gene therapy because not everybody has a match. There's some thought, although this, I will say, openly is quite controversial, that if you fix some cells, that information may be able to be shared with other cells through a process of cross-correction, although I know this is an actively debated topic on whether it actually happens. So maybe we don't need to fix every single cell, but we can just fix some cells, and they can clean up the missing pieces or the metabolites that are building up. I'm going to talk really briefly about gene therapy and the basis of it. There's something called ex vivo gene therapy, which is you take the cells from the person You give them the missing information through something we oftentimes call a vector, and then you infuse them back into the person. So you take your own cells, so it's like a bone marrow transplant of yourself, but you have replaced a missing piece. In vivo gene therapy takes the packaged information and gives it directly to the person. So it takes out that kind of autologous stem cell transplant step. We oftentimes use viruses, but if you've noticed, we don't call them viruses, we call them vectors. And that's because they're not very good at viruses anymore, as being viruses anymore, on purpose. We've basically taken out all the pieces of them that make them replicate or be able to do their own thing, and you're just left with a shell or an envelope, right? And so we use lentivirals commonly, retrovirals less so. Adenovirals were the hot thing 10, 15 years ago. We've really moved away from it because there are so many adenoviruses out there that we all can react to them and they cause a big immune response. And so the hot thing right now is adeno-associated viruses. When people say AAV, that's what they mean. The interesting thing about AAV is there are many, many different forms out there that occur naturally, and each one has different cell types that they like going to. And so rather than just using one virus and hoping it gets to where you want it to go, you can actually choose those naturally selected variants or even change it or manipulate it a little bit to make sure it goes to where it needs to go. One of the cool things that we're working on in the lab is actually using our own packaging system. So rather than using the virus as the envelope, we can package it up in something called a lipid nanoparticle. And this is cutting edge and not ready for prime time yet, but scientists are working on being able to not use a virus, but be able to use a little lipid package. I suspect as is that will get very effectively into the liver, but I think the potential is to be able to flag or tag that so you can exquisitely get that information into the cell types you want by giving it different signals on the surface. I think with the genetic manipulation of the vector or that packaging, that's where we're giving that instruction manual that's missing from our cells. And we can give that into the person that needs it through this autologous ex vivo in some. It's also important to know what types of cells or places in the body we need to get the information into. And there's a lot of controversy on route of administration. But so far, people have tried directly into the eye for eye diseases, around the liver for liver diseases, into the blood, and then various places in the brain, into the brain tissue itself, and into the variety of different fluid lakes that are around the brain. We are still, as a community, understanding the best way to give it. And it's an art more than a science as we're understanding the most efficient way to get the information to really just the cells we want to get it. Again, we have several opportunities in the leukodystrophy space, and we're really leading it as a community. We have MLD, which is approved in Europe, although there are insurance issues in Europe, so our fight is not over. ALD is approved in the US and Europe, but there are insurance issues in Europe again. So I think even once we get to an approval, the barrier isn't done. We need access for everyone. We have several leukodystrophies that have gene therapies in trial, Krabbe disease, Canavan disease, among others. And so it is a space that is going to explode in the coming years. As many people here know, there is an approved therapy using a lentiviral vector for metachromatic leukodystrophy. And that really is targeting children that have pre-symptomatic, meaning they haven't yet started having problems or very minimally symptomatic symptoms. The University of Minnesota is working on opening that early access US site. So we're working together as a community to make sure children everywhere have access to the appropriate treatments. I wanted to briefly show you a success story that is outside the world of leukodystrophies, but it's a neurometabolic disorder. In the case of AADC deficiency, children aren't able to make the enzyme that produces dopamine, and dopamine is essential for sending messages within the brain. Children have symptoms that are very similar to many of our children with leukodystrophies. They have developmental delays, motor issues, seizures. And so they had an idea that they were going to actually do direct injection of this vector, it was AAV2, into the brain to get the information to where it needed. And hopefully, I am working on my mouse. Why don't I continue? And then when I'm done, I'll try and show you the video. If not, you can go to The Lancet. There was a 2017 video. It's public, and it's where you can see how well a child who was unable to lift his head received the gene therapy and started walking again within 12 months. It's an awesome video, and I hope I can get it working. I think one thing as a community, and I'm going to ask something of you that is almost impossible to ask, is patience. Not everything we do works, and I also don't want just one option for each disease. We need to have a lot of irons in our fire if we're going to help the entire community. If we look at the lysosomal disease world, which does overlap in several disorders with the leukodystrophy world, they have been working for decades longer than us on therapy development, and they have pushed many, many things forward into clinical trial, but very few approvals. And that's because sometimes things don't work, and sometimes things worked, but we didn't ask the right question. And so we need to learn from the mistakes of our friends and colleagues, so hopefully we don't fall into the same pitfalls that have affected other people. In RNA, there are many ways, that middle step, that we can intervene as well. I love the story of how RNAi was originally discovered. It was actually an unusual petunia color, and they figured out and ultimately got the Nobel Prize in this petunia level intervention, where they found that petunias actually could modify their own color using RNA modification, so they were changing how their own colors were being made. In RNAi, your body has a naturally occurring complex that binds that mRNA. So that's the step between DNA and the protein. And that RISC complex comes in, and it has a little template in here that tells it exactly where to bind. And it can cleave, which means cuts it into two pieces. And so when the antisense is in there, you can change how the protein is being made. You can either stop it from being made, or in some cases, actually get it to skip or change those instructions. There are some approved applications. Spinraza is one. There's also an active clinical trial for Alexander disease using RNAi. This is a particularly good approach when we have splice variants, meaning you can make one form that works but not make another form that doesn't work as well, or overexpression, where things are too high and you just want to have the body make lower levels. I think the new hot area is going to be gene editing. And I think when people talk about gene editing, they talk about CRISPR, but CRISPR is really the first generation of our knowledge, and we have evolved past CRISPR at this point. CRISPR is kind of cool. It was discovered through Europe in the yogurt industry. They discovered that some of their giant tubs of yogurt lived longer, and it was because they were resistant to a bacteriophage, right? And so it was, again, another Nobel Prize that came out of a yogurt industry, which is weird. This technology is actually using all the tools that are already in our cell, and we just didn't realize that we were using these tools. Very similar to the oligo-based approaches, this also has a guide RNA that tells where an enzyme called Cas9 needs to bind. And similar, it causes a cut. Classically, what happens is it just cuts out the piece and merges it back together, but you can actually trick it into inserting a piece. So let's say you cut out the spelling error, you can put in the correct spelling, and so you would be permanently fixing the way your own cell is spelling something. CRISPR is lovely because it's cheap and easy. It makes it imminently targetable because you have that guide RNA that tells exactly where to bind. You can remove, substitute pieces of the genetic code. It's been used in Duchenne's, sickle cell disease, among others. But I think there's some problems. It's not quite as targetable as we had hoped it would be. And I think there are more elegant ways that are coming along. So let's talk briefly about what's in the future. As I said, I think there's going to be a lot more elegant ways we can get new genetic information packaged into the cells. I really want us to work on better targeting. So it's not just you are exposing the entire body to the changes, but really we figure out what cells are involved and develop exquisitely targeted therapies to help. I think we also need to work as a community on earlier interventions. That means newborn screening. That means access to genetic testing. Most of these therapies that we're talking about work best and sometimes only work if we're capturing and finding a population of children before they have problems. It is very hard for us to reverse changes. And we need collaboration between stakeholders. And I mean that between academic groups, I mean that between family groups, I mean that with industry partners. We all want the exact same thing, which is a treatment for our rare diseases. And we should learn between diseases. So in the MLD community, we have learned a ton from the ALD community, right? We know the roads that have tripped them up, and we're going to learn from it and work together as a community, and that is essential to move things forward. And the other thing I'm going to bring up, and I mentioned it earlier, is we need combinations. We treat diabetes with dozens of drugs in order to control the sugars. We treat blood pressures with multiple blood pressure medications. How can I take something as complicated as a genetic disorder that affects the brain and expect a one-size-fits-all? And so I think as a community, even once we have an approved therapy, we need more because we're still not going to be able to help everyone. And so I think working on approaches that can complement each other, maybe you get one therapy that's an oral medication. And you take that by mouth while you're waiting for your gene therapy to start working. I think there are lots of ways we can work together as a community to really reach everyone. So very briefly, the next generation is gene editing. I think we're going to be able to do things beyond CRISPR. The next hot thing is called adenine base editing, which is just a different way to target that information. It's being currently studied using lipid nanoparticles for a rare metabolic disorder, but I think that this is the kind of thing we're going to be going towards, is much more targeted therapies. Again, that nanoparticle now is pretty generic. It's just that fatty envelope, but I think ultimately we're going to be able to target that to individual cells. There's really cool, innovative work that's coming out of various groups around the country about actually doing in utero interventions. And one of the places that this is actually happening is in enzyme replacement. So as I talked about, the lysosomal enzyme disorders are really good about being able to make their enzymes and give them, but they were finding sometimes, even if you knew a family was affected, by the time the child was born, they had problems. And so people are starting to do in utero enzyme replacement under clinical trials. And so intervening before there is even a chance of any developmental injury. And there's talk about gene therapy in utero as well, although we're not there yet. I want to highlight for just a moment something called the Bespoke Gene Therapy Consortium. The FDA is listening to us as a rare disease community, that we can't have the normal pathway of a traditional phase 1 with tons of patients, going all the way through, and they're trying to build a playbook. And so the Bespoke Gene Therapy Consortium is a playbook where stakeholders are getting together and trying to find the most efficient, safest way to get gene therapy to patients with rare diseases. And so one of our leukodystrophies was actually selected for the Bespoke Gene Therapy program. And so we're excited to be able to move therapies and have a seat at the table of how we're going to get interventions to rare diseases. So in summary today, we talked about the genetics and how that information moves from DNA to RNA to protein, and how we have interventions and innovations targeting each one of these levels, from DNA with base editing and CRISPR, RNAi to targeting the mRNA level, to protein level interventions where you can give supplementation therapy and give the cells an entirely second set of instruction manuals. But if you leave this today with anything, I hope it's hope. This is a place we were not 5 years ago. This is certainly a place we weren't 10 years ago. There are incredible clinical trials happening across the leukodystrophies and innovations happening at every single step of the process. And so I have incredible hope for the future, and I hope you all do as well. So I'd like to acknowledge my place of work, which is just an extraordinary place of intervention and innovation at the Leukodystrophy Center of Excellence. We have a very large leukodystrophy team helping to research potential cures and better understanding of leukodystrophies. Many of the people are in the room today. And I also want to thank our families affected by rare diseases. This is an incredibly hard journey I can't even imagine. And having your partnership in developing therapies and being ready for clinical trials is such an important gift you are giving the world. And it is our honor to be able to partner with you as we really change the face of rare diseases together. So thank you very much for your time and attention. And I'm going to see if that video will play from here. It looks like it will not. I'd be happy to take any questions that people have. Well, far be it for me to keep you from lunch. Oh, I do have a question.
Yes, especially because lunch won't be ready for
Oh,
25 minutes.
Lunch is not ready for 20 minutes, so I apologize.
Talk away. I'm coming.
Help find additional donors or matches, or should we have family members see if they're matches? How do we go about increasing the numbers?
That is such a beautiful question. Do you mind if I repeat it just because the microphone wasn't to you yet? It was talking about the huge inequity issue related to bone marrow matches. And the answer is the National Bone Marrow Registry match, which I am not an oncologist, so I only work with them from the leukodystrophy perspective. But they have an incredible team mobilizing, and many people from our community have gone into their churches and community to get all community members to go in and enroll to reduce that inequity. But it's just numbers and people, right? I never, ever want to be faced with a situation where I say bone marrow transplant is the right way to help your child, and I still can't get there because there's not a match. That is a horrific and unacceptable answer. So enroll as many people as you can, talk to your community about Be the Match, get people to enroll. And then the other thing that we're doing from the research side is developing therapies like gene therapy that are agnostic to that, and it doesn't require a match in the community. But it's a huge issue. I lose sleep over it.
Hi. So my question is not necessarily related to the content, but I was just curious how, like at CHOP, how is your department organized, if that makes sense? Like you have a primary focus, and then like the other doctors in your office, do they have other focuses?
I think, thank you. I'll do my best to answer that. And know there are a million different ways for the care to be structured to really meet the unique needs of our leukodystrophy center. So I will share the way our program is structured, knowing that there's infinite numbers of beautiful ways that your physician's office can reach that. So I work in the Leukodystrophy Center of Excellence, and there we have a multidisciplinary care team that meets with families and personalizes the people that the family will meet with during the day to match the needs of the child, right? And so they always meet with the neurologist, and we have several leukodystrophy dedicated neurologists where really all we see is caring for children with leukodystrophies. We have a complex care physician, we have a physiatrist or someone that helps us manage tone, physical therapy, occupational therapy, speech therapy, nutrition. And then as needed, we'll bring in additional therapists such as endocrinologists to help with our adrenal leukodystrophy children, bone health specialists, GI, things like that. So it takes a village, and there are lots of different ways to organize that village.
Can you speak more about the Centers for Excellence, where they are, and how can parents access them, patients access them?
That is an incredibly good question. I glossed over it briefly, but we are part of something called the GLIA CTN. And so what this is it's a national consortium of leukodystrophy centers, and there are 8 centers around the country geographically distributed that have really that expertise to be able to care for families. And then each individual disease oftentimes has their own network of providers or doctors that they prefer and have experience in it. But I think one huge area to emphasize is we all will collaborate and help the entire country and sometimes world as much as we can. We need a network of doctors that can care for children regardless of where they live. So again, without those geographic boundaries. So I work with physicians around the world, helping them provide the best possible care they can when families can't travel to one of the specialized centers. And we're trying to train the next generation too. I teach an entire course to train pre-med students on how to become rare disease leukodystrophy doctors. So hello to all my scholars over there. Thank you. We need more. We need a bigger network. Our work is just beginning, which is super exciting. We have so many therapies in development, but in newborn screening, we are going to have a population of kids that need help in a way we aren't equipped to provide right now. So the bigger we can make our network, the better. Here's a question.
Hello? Okay. Is that on the GLIA site?
Yeah, the GLIA
The list
site.
of LCNs? Okay. Thanks.
What type of timetables are you anticipating with some of these therapies that you've been discussing?
I think, can I leave that as an ambiguous answer? It really just depends on the type of therapy, how long it takes to get through development, and where we are in the stages, right? So something like getting a gene therapy from not even having the vector or the backbone into patients in a clinical trial can take upwards to a decade. I think depending on how much we can learn from each other, we're trying to streamline that. But my ultimate goal is to get therapies approved that work and work for as many people as possible. So there are some shortcuts we just can't take. It does unfortunately take more time than we want. But I also think we should put a lot of irons in the fire. I want lots of therapies to go through, not just one. And that's because I can't always predict which ones are going to work and which ones aren't. Well, thank you so much for your patience, your time, and the beautiful questions. And I apologize for the video not working, but please check it out. It's a gorgeous video of a successful gene therapy. So thank you, everyone.